Hi guys, this is Dr. DeFeo here in my first ever video. I thought I just wanted to take a 5 minutes or so to go over some of the main points from today and to clarify some things that I was noticing that people were having trouble figuring out. First thing is you guys did a great job. The purpose of this exercise today was for you guys to engage in groups together to analyze these experiments. One of the things I want you guys to start doing, though, is when you approach an experiment, if it's a figure in this book or a figure in the future, the first thing you need to figure out is what is the independent variable, okay? That should be a signal that goes off in your head as you're thinking about interpreting the experiment. Now, my handwriting is really terrible, so I'm sorry. The other thing I want you to think about is the dependent variable. Without understanding what the experimenter is both changing and what the experimenter is measuring, it's very difficult to interpret and to understand what the conclusions are and why. So let me give you an example. Griffith's experiment. The independent variable was changing the strain of bacteria. Changing the strain of bacteria caused the mouse to, after being injected with that bacteria, to live or to die. So the thing that was being measured, of course, was whether or not, our mouse dies or lives. And that depends on whether it's the smooth strain or the R strain. Now, one of the things that Griffith experiment figured out was that there is some sort of factor that contains information that can transform bacteria. He didn't know what it was called, but he hypothesized that there was something called this transforming factor. Okay, that's all he knew. He knew that it must have contained some information because when you mix the S strain that was heat inactivated with the R strain, which is rough, he was able to turn the rough strain into a into a bacteria that can kill the mouse. Okay? Previously couldn't kill the mouse, but once you take the contents from the heat-killed S strain into that and mix it with the R strain, you can turn it into a bacteria that can kill the mouse. So what is it that the S strain, that heat kill, what is that heat kill? So if you have this bacteria, of course, you know that there are four macromolecules. You need to remember that you have DNA, we talked about inside here, you have RNA, you've got proteins all over the place, you've got lipid, and you've got sugars. You have all of this stuff. Which one of those molecules? It's a molecule that's doing it. Which one is going to be the one that turns the R strain into it? Griffith didn't know it. All he knew is that one of those molecules need to be it because he was able to transform or change the R strain into a strain that can infect and kill the mouse. Avery is the one that came around and figured out that transforming factor can be DNA. And he figured that out because he treated, he took that bacteria and added heat, which when you add heat to a bacteria, essentially what you do is you end up blowing it up. Okay? So the only thing that's left over is biomolecules. And then what he did was he added enzymes that can break down I'll actually say destroys. Let's just say destroys so that you guys can remember that the enzyme can destroy protein, lipid, sugar, RNA, and DNA. And when he tried to transform by taking this soup, okay, and then mixing it with live bacteria, if he added the enzyme that destroyed the protein, it still transformed. If he destroyed the lipid, you were still able to transform the bacteria. If you destroy the sugar, you were still able to transform the bacteria. If you destroy the RNA, you were still able to destroy the you were still able to transform the bacteria. But if you destroy DNA, now the bacteria no longer gets transformed. And so Avery was able to conclude that the transforming factor needed to be DNA. But that doesn't demonstrate that in the initial experiment, that doesn't necessarily. There were people that were so convinced that protein was the transforming factor that it wasn't enough for some people. They're like, that's great, but maybe there's something that we don't know going on. You always have to contend with that in science, and that's why you need to constantly validate your experiment. And that's what Hershey and Chase did. Hershey and Chase manipulated the experiment and measured something. So the first thing you need to think about is, what's the independent variable? The independent variable is the change. The independent variable is how about this? The independent variable is changing the radioactive isotopes that the virus was grown in. So you can imagine that when you're starting your experiment, you have your virus and you need to grow it in media. And in one media, or in one culture, you had P32, and in the other culture, you had S35. So when you put the virus in P32, you're able to label specifically the DNA. When you grow your virus in S35 media, you're able to make lots and lots of virus that now have protein that is covered in radioactive sulfur. Why is this important? It's so critical because phosphorus is only found in DNA or RNA, but remember, we know that it's not RNA that's a transforming factor, because it was only the enzyme that broke down DNA that prevented transformation. And sulfur is only found in protein. So when they did this, you need to say, well, what's the dependent variable, Dr. DeFeo? Well, the dependent variable is measuring radioactivity. So you can imagine making your graph, and you have radioactivity going on your y-axis. Now you're changing on the x-axis. So let's say you do this experiment, okay? And in your first experiment, you label the DNA with the phosphorus. Then you measure the amount of radioactivity in the bacteria after removing all of the virus, and you see, whoa, you have a big signal with P32. But when you do the same transforming operation, it's not that this virus wasn't able to transfer its genetic material in. It's still able to transfer its genetic material in. It's just that because you grew it only in S34, that is washed away when you remove the virus. So when you do your not S34, but S35, you get no radioactivity. And so from this experiment, you can conclude that since the P32 gave you a signal in the bacteria that was infected with the bacteriophage, that the only way that could happen is if the genetic material itself was labeled with the P32 and that it was injected into the bacteria, and helping to demonstrate definitively that the genetic material that can be passed into the bacteria is DNA. And that's where the information comes from. So DNA, and kind of in summary, DNA stores information. Okay, so you can open that book. We're going to learn about that tomorrow. You can have ion movement, ion transport, movement, cell growth, respiration, or it can kill bacteria, right? I'm sorry, it can kill mouse if you're a bacteria. So all that information is stored in your genome. Then you need to also copy and you need to transmit it. We know it can be transmitted because that's what we're doing, right? Where in this case, where we can transform bacteria, so we can move DNA across the membrane and into a cell. Now, that's of course not how organisms reproduce. Viruses can transmit it. We're going to learn eventually how, we learn through mitosis anyways, that you can make copies and transmit. And we're going to eventually learn how in meiosis, how we can actually make sex cells that eventually will turn into another living organism. So that was my first go. I hope that is somewhat helpful for those of you who missed some of these points. But I think the most important thing is actually this right here. DNA stores information. DNA needs to be able to copy information and transmit it. How does it do that? We'll learn that tomorrow.